My name is Alka Saxena, and I am the head of the Genomics Research Platform and Single Cell Laboratory at the NIHR Guy's and St. Thomas's Biomedical Research Center. In my lab, we are helping researchers with conducting single-cell genomics investigations on a diverse range of projects, from Crohn's disease and peanut allergies to cell therapies and immunotherapy. 10 years ago, as an international fellow at RIKEN in Yokohama, Japan, I was investigating Rett syndrome through systems biology approaches. Rett syndrome is a neurodevelopmental disorder that affects mainly girls. 80% of patients with Rett syndrome have a mutation in the MECP2 gene, which is located on the X chromosome. In females, each cell carries two X chromosomes, one inherited from the father and the other from the mother. The mutant gene can be either on the paternal or the maternal X chromosome. The gene is a transcriptional regulator. Therefore, the transcriptomes of cells that have the mutant gene as the active X chromosome would have a different transcriptional profile than the cells that have the active X chromosome that carries the normal gene. So it was apparent to me that I needed to do transcriptional profiling of individual cells to be able to find transcriptional differences between cells that had the mutant gene compared to those that had the normal gene. So I was on the lookout for technologies that allowed single-cell transcriptome analysis. In 2015, I was given an opportunity to participate in an MRC funding call that allows setting up of infrastructure for single-cell genomics. Single-cell technologies are becoming easier and easier, and therefore they are becoming more accessible. And as awareness grows, I wouldn't understand why any researcher would want to conduct their analysis on bulk level when single-cell approaches are available to answer the same question. The considerations that researchers need to give thought to are to match the technology with the research question, whether high-throughput shallow sequencing would suit their research question better, or do they need to deeply profile cells. Consider optimizing upstream workflows. That is really important. I cannot stress enough the importance of this. Consider read depth. If they don't have enough number of cells, they need to consider cell hashing approaches where they can mix samples together and still conduct high throughput investigations. My advice to researchers who are starting to think about single cell experiments is to spend a lot of time on upstream processes. The quality of cells that goes into your single cell experiments is key to getting good data. Too often we find suboptimal cells being provided to us for these studies. Single cell suspensions that are free of debris and contain high quality cells and viable cells are essential for these analyses. My advice to researchers considering single cell investigations is to learn bioinformatics analysis. Having these bioinformatics skills is no longer a luxury, and it is much easier than it looks. There are software programs available to download and to purchase that follow all the highest standards that are recommended by the experts in single-cell genomics. Vendors that manufacture instruments for single cells also provide software programs to download and conduct data analysis. Most of these programs are based on graphics user interface, the GUIs, so the data is analyzed through pointing and clicking. And I would strongly recommend that researchers who know their research best conduct their own data analysis and learn these skills, which have become a necessity. The current trend in single-cell genomics is towards multi-omics analysis. Perturb-seq and site-seq, reap-seq and cell hashing, transcriptome sequencing along with VDJ sequencing. All of these technologies provide great insights into the biology at a single-cell resolution. The added layer of information provided by multi-omics analysis really provides novel insights and clarity at a single-cell resolution. For example, overlaying VDJ clonotype information on single-cell transcriptomic datasets from T cells allows us to find subtle clonotype-specific differences between T cells that may be clustering together. The researchers at RIKEN sequenced single cells using a modified method where they used random primers which enabled capturing of stranded 5' ends of individual cells. Upon sequencing these cells, they found non-coding RNAs, but they focused on enhancer RNAs. Now, from bulk studies, we know that enhancer RNAs are bidirectional RNAs expressed from enhancer regions. Upon single-cell analysis, they found that at an individual cell level, these RNAs are unidirectional, which is a great insight that could only have been provided through single-cell investigations. What I would like to see in single-cell genomics is profiling the entire length of the transcript with strand information conserved on short-read sequences. In addition, I would like to be able to capture non-coding RNAs, microRNAs, together with long RNA. Additionally, combined investigations of the genome with transcriptome at a high throughput level, and combined investigations of accessible chromatin along with transcriptome information will provide us information on genetic and epigenetic drivers. That is what I want to see in single-cell genomics. The Biomedical Research Center at Guy's and St Thomas is funded by a grant awarded by the National Institute of Health Research. These grants are awarded every 5 years, jointly to a hospital and university. In our case, that is Guy's and St Thomas's and King's College London. This award enables the BRC to establish world-class infrastructure and forge partnerships between clinical and academic experts. The aim of the BRC at Guy's and St Thomas's is to accelerate translational research and scientific discoveries using cutting-edge technologies. The aim of the Genomics Research Platform is aligned with the aim of our BRC. We aim to resolve research and clinical questions through the application of high-resolution leading-edge genomics methods. As we move towards an era of personalized medicine, it is important to catalog the transcriptional output of each cell to understand normal states from abnormal states. This would not only help us in finding transcripts that are misregulated and finding biomarkers for therapy, but also allow us to understand treatments at an individual cell level to better design targeted therapies.